Welcome to the deep dive. Today we're tackling a really specific area of forensic biology. How experts identify two crucial types of bodily fluids at crime scenes, vaginal secretions and menstrual blood.
Yeah, and it's more than just linking someone via DNA. Identifying the type of fluid gives us what we call probative value. It helps confirm or refute specific claims about what happened.
Probative value, right. So finding DNA is one thing, but knowing if it came from, say, vaginal fluid versus just sweat or skin cells, that can completely change the story in court, especially in sexual assault cases.
Exactly. It can corroborate an allegation of a sexual act versus a claim of just, casual contact. So our mission today is to unpack the science behind making those distinctions.
Okay.
We'll start with vaginal secretions, looking at cells, chemicals, the whole deal. Then we'll shift gears to menstrual blood and how it's different from, say, blood from a cut.
All right, let's dive in. Vaginal secretions first. Starting at the cellular level, what makes these epithelial cells distinct? They're not like skin cells, are they?
No, not quite. They are stratified squamous epithelial cells, but the key difference is they're non-keratinizing. Skin cells have that tough, keratinized outer layer that constantly sheds. Vaginal cells don't.
Okay, non-keratinizing. But what's the specific visual cue scientists look for?
It's what's inside. Glycogen. Lots of it, especially in the intermediate and superficial layers of the cell's cytoplasm. Think of it as stored energy.
Glycogen. So these cells are just packed with this carbohydrate. Is that always the case?
Well, it's heavily influenced by estrogen levels. So in women of reproductive age, you'll typically find abundant glycogen, but before puberty or after menopause, estrogen is lower, and you see more parabasal cells, which don't stain as strongly for glycogen.
Interesting. So if the glycogen is there, how do forensic scientists actually visualize it? You can't just see carbohydrate under standard microscope.
Right. You need a stain to make it pop. The classic method, the sort of go-to for years, has been Lugol's iodine staining.
Lugol's iodine, how does that work?
It's basically iodine made soluble with potassium iodide. The neat part is the chemistry. The iodine molecules, specifically triiodide ions, fit snugly into the helical structure of the glycogen polysaccharide.
Like a key in a lock.
Kind of, yeah. And when they bind, they form this dark brown glycogen-iodine complex. It's really visible.
So that makes the glycogen stand out. Is that the only way? I think I've heard of another one, PAS.
Yes, the periodic acid shift method, or PAS, that's another option. It stains the glycogen in the cytoplasm magenta and the nucleus purple. So different colors, same goal. Highlight the glycogen.
Okay, sounds straightforward enough. Stain for glycogen, find the cells. But I suspect there's a catch.
There always is in forensics, isn't there? The catch is specificity. Glycogen-rich squamous epithelial cells aren't only found in the vagina.
Ah, where else might you find them?
You find them in the mouth, the buccal cavity, also the pharynx, esophagus, anus, and even the urethra.
So finding these cells doesn't automatically mean vaginal secretions. And I bet the buccal cells look pretty similar.
Worse than similar. Buccal cells and vaginal cells are morphologically indistinguishable under the microscope using these basic stains. You just can't tell them apart by looking.
Right. That's a problem. So finding glycogenated cells isn't definitive proof. How do we get past that hurdle?
We need a more sophisticated staining approach. That brings us to Dane's staining method. It's a differential stain designed specifically to tackle this problem.
How does Dane's method work differently?
It uses a combination of stains after fixing the cells with methanol. Instead of just looking for glycogen, it produces distinct color profiles for the different cell types. Skin cells end up looking red or orange. Buccal cells look orange-pink, but crucially, their nuclei stain red.
And vaginal cells?
Vaginal cells stain a characteristic bright orange, and importantly, their nuclei also appear orange. So it gives you these distinct color signatures to differentiate between skin, buccal, and vaginal epithelial cells. Much better specificity.
Okay, that makes sense. A visual way to tell them apart. Now, moving beyond the cell structure itself, what about chemical markers within the vaginal fluid? Enzymes, maybe?
Yes, exactly. One potential marker is an enzyme called vaginal acid phosphatase, or VAP. It's normally produced in small amounts by cervical epithelial cells.
Acid phosphatase. That sounds familiar. Isn't there another acid phosphatase important in forensics? From semen?
You got it. Prostate acid phosphatase, or PAP. It's a major biomarker for semen, usually present in much, much higher quantities than VAP in sexual assault evidence.
Okay, so if you have a sample with both semen and vaginal secretions, you've got VAP mixed with potentially huge amounts of PP. How do you tell them apart if they're both acid phosphatases? Are they chemically different?
That's the challenge. They're incredibly similar. Same molecular weight, same enzymatic activity, they even react the same way to inhibitors. Chemically, they're like twins.
So chemical tests won't separate them.
Yeah.
How do you isolate the VAP signal then?
We turn to physics, specifically agarose electrophoresis. We use an electrical field to make the molecules race through a gel.
Right, electrophoresis separates things based on charge and size.
Exactly. And it turns out PAP has a slightly higher electrophoretic mobility than VAP. It moves faster towards the positive electrode, the anode.
Ah, so PP pulls ahead in the race.
Precisely. Even though they're chemically similar, that subtle difference in charge or shape lets them separate physically on the gel, so we can detect VAP on its own even if it's swamped by PAP.
Clever. Using physics when chemistry hits a wall. Okay, what about other biological markers, like bacteria? The vaginal microbiome is pretty distinct, isn't it?
It is. We often look for DNA from Lactobacillus species like L. iners or L. crispatus. These are rod-shaped, gram-positive bacteria that dominate the healthy vaginal microbiome and produce lactic acid, keeping the pH low.
So can we use 16S RNA gene sequencing to find Lactobacillus DNA and confirm vaginal secretions?
We can certainly detect the DNA, yes, but again, we run into a specificity issue. Lactobacillus isn't exclusive to the vagina.
Oh, where else?
They can sometimes be found in female urine samples, just due to the proximity of the openings. And, somewhat confusingly, they can occasionally be detected in semen as well.
So finding Lactobacillus DNA is suggestive, maybe points you in the right direction, but it's not definitive proof on its own.
Exactly. Not strong enough to stand alone in court as confirmation of vaginal fluid.
Okay, so the staining has limits, the enzymes need careful separation, the bacteria aren't specific enough. This feels like we're leading up to a more definitive method.
We are. For true confirmation, especially when dealing with potential false positives or negatives from older methods, we look at RNA. Specifically, mRNA, the messenger RNA.
Ah, the active genetic blueprint. So instead of the cell structure or protein, you're looking at what genes are actively being expressed by that tissue.
Precisely. We use a technique called reverse transcription polymerase chain reaction, RT-PCR, to detect mRNA markers that are highly specific to vaginal tissue.
What kind of markers are we talking about?
Two key examples are MUC4 and HBD1. MUC4 is a gene that codes for a major mucin protein found in vaginal mucus. HBD1 codes for an antimicrobial peptide, beta-defensin 1, which is also characteristic. Finding the mRNA for these means the cells were actively producing these specific vaginal components.
And these mRNA markers aren't typically found floating around in other body fluids.
Generally, no. Their expression is highly tissue-specific. We're also seeing the use of microRNAs, like miR-1214, adding another layer of confirmation. RNA analysis gives us that really high level of certainty.
Okay, that provides a strong confirmation for vaginal secretions. Let's switch gears now to blood, specifically distinguishing menstrual blood from peripheral blood. Why is this distinction so important in, say, a sexual assault case?
It's critical. If blood is found, the defense might claim it's from an incidental injury, a scratch, a nosebleed, something unrelated to the alleged assault. But if forensic analysis confirms it's menstrual blood, that points much more strongly towards contact related to the complainant cycle and potentially corroborates aspects of their account.
So what makes menstrual blood biologically different from regular blood coming from a vein or a cut?
It comes down to the whole process of menstruation. It's not just blood, it's the shedding of the uterine lining the functionalis layer of the endometrium. This layer is rich in specialized spiral arteries.
And the shedding process itself changes the blood.
Dramatically. When progesterone levels drop, these spiral arteries constrict, cutting off blood flow that's ischemia. This leads to tissue death, hypoxia, and importantly, widespread enzymatic breakdown of the endometrial tissue. Menstrual fluid is a complex mix of blood, degraded tissue, and lots of enzymes.
Enzymes, that sounds like a potential marker. Is there something unique about blood clotting in this context?
Absolutely. The uterus has a really fine-tuned system called endometrial hemostasis. It needs to clot blood where vessels break, but it also needs to break down those clots rapidly, a process called fibrinolysis, to prevent the uterine cavity from filling up with large clots during menstruation.
So it's clotting and dissolving clots at the same time, almost?
In a highly regulated way, yes. And this intense fibrinolytic activity, the clot busting, leaves behind a very specific molecular signature.
And that signature is what forensic tests look for. What is it?
The key marker is D-dimer. When a blood clot forms, fibrin proteins cross-link to stabilize it. Then an enzyme called plasmin comes along during fibrinolysis to break down that stabilized fibrin. D-dimer is a specific fragment, a degradation product left over when plasmin chops up cross-linked fibrin.
So, because there's so much clot breakdown happening during menstruation,
You get very high levels of D-dimers in menstrual blood, much higher than you'd find in peripheral blood from a simple injury.
how do labs test for D-dimer?
There are a few methods, like ELISA or latex agglutination, but a common one used now is the immunochromatographic assay. Think of it like a rapid strip test.
Fast and easy.
Relatively, yes.
Yeah.
They're very sensitive and specific. Using monoclonal antibodies designed to bind only to the D-dimer fragment, you get a result in minutes.
And crucially, peripheral blood doesn't trigger a positive result.
Correct. While trace amounts of D-dimer might be in peripheral blood from normal bodily processes, it's generally far too low to give a positive signal on these specific forensic assays. So a positive D-dimer test is a strong indicator of menstrual blood. We should note postmortem blood can also have high D-dimers, but that's rarely a factor in typical sexual assault scenarios.
That's a powerful differentiator. Okay, are there other markers besides D-dimer? Enzyme-based ones, maybe?
Yes, another classic approach involves looking at lactate dehydrogenase, or LDH. LDH is an enzyme found in many tissues, involved in energy metabolism. It's a tetramer, meaning it has 4 subunits.
And there are different versions of LDH.
Exactly. There are 5 main isoenzymes, LDH1 through LDH5, made up of different combinations of 2 subunit types, A and B. Different tissues have different patterns of these isoenzymes.
Ah, so it's not just the presence of LDH, but the pattern of the different forms. How do you see that pattern?
Again, we use electrophoresis. We separate the 5 LDH isoenzymes on a gel based on their charge.
And the pattern differs between peripheral and menstrual blood.
Significantly. In peripheral blood from an injury, you typically see LDH 1, 2, and 3 as the most prominent bands.
Okay.
But in menstrual blood, the pattern consistently shifts. You see a clear predominance of LDH4 and LDH5. This distinct isoenzyme profile is another reliable indicator used to identify menstrual fluid.
Interesting. So, two different approaches, D-dimer focusing on clot breakdown, LDH on enzyme patterns, both pointing towards menstrual blood. Now, just like with vaginal secretions, is there an RNA-based confirmation for menstrual blood too?
Yes, and arguably it provides the highest specificity. We again look at mRNA expression using RT-PCR, this time focusing on genes involved in that tissue breakdown we talked about.
The breakdown of the endometrium, what kind of genes are those?
We look for matrix metalloproteinases genes, or MMPs. These code for enzymes, endopeptidases, whose job is basically to chew up the extracellular matrix, the scaffolding, holding the endometrial tissue together. They are crucial for the controlled destruction during the menstrual cycle.
Makes sense. Are there specific MMPs that are the best markers?
MMP7 is one marker that's looked at, but the real standout, the most sensitive and specific one currently, is MMP11.
MMP11. What makes it so good?
Its expression pattern. Using RT-PCR, you can detect MMP11 mRNA reliably in menstrual blood, typically from day 1 through day 8 or so of menstruation.
And the key question, is it found in other fluids?
Crucially, MMP11 mRNA is generally absent in peripheral blood samples and also in vaginal secretions. That makes it an incredibly powerful, specific marker for confirming that a bloodstain is indeed menstrual in origin. Of course, like any marker, there are potential caveats. MMP expression can sometimes be elevated in other situations, like wound healing or certain cancers, so results always need careful interpretation and context, often using multiple markers. But MMP11 is a cornerstone of modern menstrual blood identification.
Wow. So across both fluids, we've gone from basic staining and cell morphology right up to highly specific RNA signatures. It's quite a journey. We saw Lugol's iodine lighting up glycogen, electrophoresis teasing apart VAP and PPA based on speed, D-dimer acting as a fingerprint for clot breakdown, that LDH pattern shift, and finally MMP11 mRNA as a near definitive confirmation for menstrual blood.
It really highlights the layers of analysis involved. And what I find fascinating is how fundamental biological processes like maintaining pH with bacteria or the body's intricate system for controlling bleeding and clearing clots in the uterus inadvertently create these unique biochemical trails.
Trails that forensic science can then follow.
Exactly. The very mechanisms the body uses to function normally leave behind the specific evidence needed to reconstruct events in a forensic context.
That leaves a really interesting thought for you, our listeners, to ponder. Think about all the complex, everyday biological processes happening in our bodies. How many of them are leaving behind unique, traceable signatures that science just hasn't fully decoded or applied forensically yet? The potential seems huge.
It certainly makes you think about what future capabilities might emerge.
Absolutely. Well, thank you for walking us through that complex science. And thank you all for joining us on this deep dive. Keep questioning, keep learning, and explore the science behind the evidence.